All right, let's jump right in. Today we are tackling one of the biggest challenges in modern genomics, finding that one tiny disease-causing needle buried in the massive haystack of our genetic code.
I mean, think about it. Our sequencing tech is so incredible now that we can map out a person's entire genome. But the flip side is that you get this absolute tidal wave of data. So the real question becomes, how on earth do you find that one critical variant causing a rare disease when you're staring at thousands of them?
Well, a lot of the time, the culprits we're hunting for are something called structural variations, or SVs for short. Now, these are not your tiny single-letter typos. No. Think of it like this. It's like whole paragraphs getting deleted from the book of life, or entire chapters being duplicated, or pages getting jammed in where they just don't belong. And these big, large-scale changes are a huge source of genetic disease.
And just to give you a sense of the scale we're talking about, get this. A typical person has more than 4,000 of these structural variations in their genome. 4,000! That creates this enormous genomic haystack, and our job is to find that one single pathogenic needle.
So you can see the problem, right? For a clinician, for a doctor trying to get a diagnosis, sifting through that mountain of data is just an incredibly difficult and massively time-consuming job.
But what if you had a specialist on the case, a kind of genomic detective? Well, that's exactly where this tool comes in. Meet our protagonist, AnnotSV. It's a bioinformatics tool designed to do exactly this.
You can really think of AnnotSV as that detective. It takes the huge list of SVs from a patient and it just goes to work, systematically investigating every single one, gathering clues, and basically building a case file on each one to figure out if it's the bad guy.
And the way it works is actually pretty elegant. Step 1, you feed it the raw data. Step 2, the engine starts its investigation, pulling evidence from dozens of different sources. And then, Step 3, it gives you a clean, ranked, interactive report that a clinician can actually understand and use.
So how does this detective actually do its job? Let's pop the hood and take a look inside the toolkit.
First off, the sheer depth of the background check is just incredible. We're not talking about looking in one or two places. AnnotSV cross-references more than 20 different scientific databases to pull together clues for every single variation. It's incredibly thorough.
And what kind of clues is it looking for? Well, the whole shebang. It checks if the variant is in a neighborhood already known to cause trouble. It looks at scores that tell you how much a gene hates being disrupted. It even looks for those tricky regulatory elements, the DNA switches. And it checks population data to see how rare it is. Crucially, it ties it all back to the patient's actual symptoms.
Okay, so you've gathered all this evidence. Now what? You can't just hand over a massive file. The detective's real job is to identify the top suspects, and it does that with a really powerful ranking system.
This is how it does it. It uses a super clear 5-class system that's totally compliant with the official guidelines from the American College of Medical Genetics. It sorts every SV into a class, from class 5, definitely pathogenic, all the way down to class 1, totally benign. This gives doctors a clear, confident place to start their investigation. It's a game changer.
Now, this next part is seriously clever. It's like checking a suspect's alibi. So imagine a sequencing tool claims that a gene is completely gone, a total deletion. And AnnotSV then does its own digging. It scans the rest of the patient's genetic data. If it finds little telltale signs, these heterozygous variants, that prove 2 different copies of that gene must exist, then ding, 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 the original claim was a false positive. It's a brilliant internal consistency check.
All right, theory is one thing, but let's talk about the real world. This is where we see how this tool actually solves real medical mysteries and changes lives.
So let's look at this case. A patient with a condition called Rieger anomaly. The sequencing found a huge deletion on chromosome 4, almost 1 million base pairs long. Seemed like an open and shut case, right? But there was a big problem.
Here is the thing that had everyone scratching their heads. The deletion didn't actually touch any genes that make proteins. From a standard point of view, it looked like it didn't do anything important. The clinicians were completely stumped.
And here is the aha moment, because AnnotSV looks at everything. It found that while the deletion missed the gene itself, it wiped out the critical genetic switches, the enhancers, that turn that gene on. And those switches were located almost 200,000 base pairs away. It was the smoking gun. Case solved.
And this wasn't just a one-off. In another really high-stakes prenatal case, a massive deletion was found. AnnotSV immediately and automatically ranked it as class 5 pathogenic.
This gave the family and their doctors a fast, confident answer when every single moment counted.
So this is already incredibly powerful stuff. But the field is moving so fast. What's next for our genomic detective? Where does it go from here?
Well, as our ability to read DNA gets better, the tool gets smarter. It's constantly being updated to handle data from new tech, like long-read sequencing, which gives us a much clearer view of the genome. It's also adapting to use brand-new pan-genome references, which better reflect the amazing diversity of human genetics. The detective is always learning new tricks.
So, what's the bottom line? By automating the incredibly tedious and difficult work of interpretation, tools like AnnotSV are freeing up our amazing geneticists to focus their brainpower where it's needed most, on solving the toughest cases and pushing us all toward a future of truly personalized medicine.
And that kind of leaves us with one last big question to think about. As we get better and better at mapping the full spectrum of human genetic diversity, the entire pangenome, what other hidden instructions, what other secret languages written in our DNA are we finally going to learn how to read?